Hi, my name is Jonathan. I'm a tutor on Chegg Tutors, and this lesson is about two-point test crosses in genetics, which is used to analyze the recombination frequency between two linked genes. Now, linked genes just means that they're close together on a chromosome, so they have the potential to stay together during meiosis and the crossover events that take place during meiosis. So they won't give you that classic Mendelian independent assortment progeny. Instead, they'll have these weird numbers that we'll take a look at. So we do these two-point test crosses by crossing an F1 individual with a homozygous recessive individual for both genes. And we cross it with a homozygous recessive individual for both genes because, as we'll see, we can kind of neglect that entire genotype because that individual can only have one type of gamete, so we don't have to worry about it. And we usually do these test crosses. We form the F1 progeny by mating two purebred individuals for both genes. So one will be homozygous dominant for both genes. So, for example, big A, big A, big B, big B. And the other one will be homozygous recessive for both genes. So little a, little b, little b. Let's take a look at two made-up genes in flies. So A and B. Big A is dominant to little a, and big A results in long bristles, which will be our wild-type phenotype. And little a results in short bristles, which will be our mutant phenotype. Big B is dominant to little b, and big B results in red eyes, which will be our wild type, and little b results in white eyes, which will be our mutant. Now let's take a look at crosses done with these two genes. Let's say we cross two parents. One will be homozygous dominant for both genes, like I mentioned before. The other will be homozygous recessive for both genes. Now, this parent can only make little a, little b gametes. This parent can only make big A, big B gametes, so they come together to make your F1 progeny, and all of which are heterozygous. Now, in order to do our two-point test cross, 2 points meaning two genes, and test cross meaning crossing the F1 with a homozygous recessive for both genes, do that. So this individual can only make gametes little a, little b. And if we were to assume that these genes are not linked and assort independently according to just classic Mendelian genetics, this individual can make gametes A, big B, little b, big A, little b, and little a, big B, and all of which will be at an equal probability. So we'll have 25% of this, 25% of this, and so on. Now, since all of the F2, the progeny that's formed between this cross that we're doing right now, they will all have this little a, little b gamete or chromosome, no matter what. So we don't even really need to worry about that for our genotype and phenotype purposes of F2, so we can kind of just neglect this because they'll all have it, and it doesn't really do anything because it's recessive alleles. So we can just look at basically this for our genotype of the F2. So F2, that's the great thing about test crosses, that we don't need to worry about the gamete from this individual. Homozygous recessive for both genes. So this is what we would expect if it is sorted independently. And let's say we had a total of 1,000 F2 progeny, and remember we had 25% of each genotype. So our big A, big B, we would have 250 progeny of that. Little a, little b, we had 250 out of 1,000. And big A, little b, we would have 250, and so on. But now let's look at some data if these genes are linked. Okay, so here we have 400 big A, big B, 400 little a, little b, 100 little a, big B, and 100 big A, little b. How do we explain this? Well, the only explanation, because we're not getting this classic Mendelian progeny, is that these genes are linked, meaning that they're close together on a chromosome. So let's kind of backtrack here and look at the parents that form that original F1 progeny. So we had this cross to form the F1 heterozygotes. This individual's chromosomes look like this. if they were linked, if these genes are close together on a chromosome, whereas this one looks like this. Now, if those two gametes come together, and the two chromosomes come together, all our F1 chromosomes would look like this. Remember, these are homologous chromosomes. So if they're linked, meaning that these genes are close together on the same chromosome, during any crossover events that take place in this F1 progeny when they form gametes during meiosis, these genes are going to stay together. So let's say we have a crossover event here and here and here and here. These 2 points break off and they flip-flop, and you still get left with one chromosome that has little a, little b, and one chromosome that has big A, big B. So a majority of the progeny will have one of these chromosomes. And that's why we're seeing a majority 400 big A, big B, and 400 little a, little b, because these two genes stay together during crossover events in meiosis. But a small amount of time will have crossover events that take place between the genes, like this. And in this case, we'll have this as our final result. But this only happens a small amount of time because A and B are so close together that the probability of a crossover event taking place between the genes is relatively small, but it also tells us something about the relative distance between those genes. So a higher recombination frequency between those genes means that they're further apart. A smaller recombination frequency means that they're closer together, and we don't have many crossover events that take place between those genes. So like I just said, we can look at the recombination frequency and establish the relative distance between the genes. So here are our parental genotypes where crossovers did not take place, and we get left with the same. Remember, this is our F2 after the F1 was test crossed with a homozygous recessive individual, so we don't need to worry about that little a, little b chromosome. So these are our parental, where crossover did not take place, and these are our single crossover events right here. So we can see that a single crossover event took place 100 plus 100, so 200 times out of 1,000 progeny. So we can get the percentage of this, so 200 divided by 1,000 times 100 is equal to 20%. So this tells us the recombination frequency. What is the percentage that these two genes recombined, which is 20%. So we can make this into an equation by saying the number of crossover events divided by total progeny times 100 equals percent recombination, or the recombination frequency. Now, the recombination frequency, like I said before, also gives us the relative distance between these genes. And in genetics, we label this as centimorgans or map units. It's hard to translate this into a physical distance because it's just relative to something else. So if we have a 20% recombination frequency, this is equal to 20 centimorgans, 20 centimorgans, or 20 map units. So that means that genes A and B are 20 centimorgans away. So in sum, we take the F1 progeny, test cross it, look at our F2 progeny, and we look for the most abundant, which will be our parental genotypes. And then we look for our crossover events, and we take the crossover events divided by the total progeny times 100, which will give us our percent recombination, which also tells us what is the relative distance between these two genes. So I hope this helped, and my name is Jonathan, and I'm on Chegg Tutors.